the methods database is something that's been going on as a loose collaboration between Allotrope and Pistoia for a number of years, but have, I think, some really interesting and exciting results and things to update us on here. And so the first presenter will be Bertha Nielsen, who is from the Pistoia Alliance and heads up their life sciences and biotechnology group there. The second presenter will be Asadin Dabo, who does method development and in silico modeling from GSK. And then finally, the third presenter for this presentation will be Pankaj Agarwal, who's Associate Principal Scientist at Merck. And from there, I think we're all set. You can go ahead and take it away.
Brilliant. Thank you. Thank you for the introduction. And it is indeed a great pleasure to be able to present again at the Allotrope Connect meeting to give you an update where we are with the methods database project. As you said, it is a collaborative project under the Pistoia Alliance legal framework. To the ones that doesn't know the Pistoia Alliance, it is a global not-for-profit members organization with the aim to collaborate to lower barriers to innovation in life science and healthcare R&D. And I think we have now more than 200 members across the entire life science ecosystem, the top pharma companies. We have patient-led research groups, we have technology and solution providers, we have academic institutions and so on, publisher. And we indeed have this unique framework where we can collaborate to enable all our organizations to work together to solve common problems and lower barriers. So the team here, we presented at one of the previous Allotrope Connect meetings, and we've shown what is the vision for the methods database. And at the last meeting, we presented Heiko from Agilent, Wolfgang from Sontal, showed you what is the technical solution. And so today, Pankaj and myself, I'm not sure if Asadin managed to make the meeting, but if not, then Pankaj and myself, we're going to show you how we have now executed this vision. And we will show you some data from the implementation at Merck and GSK and show you how we have successfully been able to transfer methods. I'm not sure my slides are changing. Ah, there they are. So yeah, so as I said, a collaborative project, and of course, you know, really grateful to all the participants in this project, Agilent, Merck, GSK, Sontal, who have really done a tremendous amount of work to help drive this project forward. So I think, you know, what is the methods database about? What is the problem statement? I think it was really well articulated yesterday as well in one of your presentations. We have to transfer data or analytical methods, in this case, consistently across the different business units, internally, externally. And this has been a challenge, and it is still a big challenge. And scientists, they mainly do this through manual file transfer in many different formats or descriptions. And that means that the scientist receiving these methods needs to completely rebuild them. And that's not very efficient. And these method descriptions, they might be extremely detailed, but reproducibility really decreases because they are text-based, but also because we use a lot of different terminologies, and this just opens up for human interpretation errors. And there is also a lot of different versions, and of course you need to make sure that you're executing the right version. So you see how all of this compiles to quite a substantial problem. And of course there are all the different proprietary platforms that really prevents interoperability. And I think we heard this as well, you know, we need to make sure that we have some sort of local storage or central storage for, not local, but central storage for these methods, you know, otherwise we are at risk of losing them in case of cyber attacks and so on. So what the methods database project is really about is to try to move away from these text-based methods. We want to have method descriptions that are fully digital, that are completely machine readable, and we want to not only improve the automation side of things, but we want to make sure that the methods can be transferred interoperably so that we can optimize efficiency and reduce cost, improve quality. So the methods database project started back in 2018, and the group built a data model and ontology to support HPLC UV methods. So we chose HPLC UV methods because, or high performance liquid chromatography, and I should probably say that this is a technique that scientists use in pharmaceutical companies to separate and identify and quantify their components in mixtures and formulations and so on. And the reason why we're focusing on HPLC is the majority of analytical data is HPLC UV data, with more than 10,000 new instruments sold each year. And you see a very simplistic illustration here of a HPLC system. And we have a lot of parameters that are easily standardized. You have, for example, the solvent that you are pumping through the system. So you need to describe the mixture and the flow rate you are pumping it through with, and your the column, the injection volume, and the settings for the UV detector. And all of this is as I said, it's fairly easily to parameterize because things comes with a quantitative value and a unit. And so the team then had all these parameters defined and built this semantic data model. The UV, the LCUV results are really influenced by a combination of multiple methods parameters. So the group selected the RDF graph representation of the methods to standardize the terminology across the vendor systems, but also to model the relationship between the method parameters. So the next phase of the project was really to get the vendors on board and to build RDF adapters or converters to the different instrument control systems. So here you see we have like a water Empower system, and we will have an adapter now that allow you to download the method from a waters instrument or water control CDS system, upload it into the methods database, which is in the Thornhill space, and then you can then download this method, convert it from ADF into something that Agilent Open Lab CDS system can understand and execute. With the methods database here, we have the ability to search, to report, and Pankites will show you how we can overlay different results files as well. So at the moment, we have around 56 parameters that describes the HPLC method, and this is more than enough to transfer these descriptions. We can link it to a LIM system, and which you see here, and you will send your sample information. So you send it from your LIMS or your ELN or whatever workflow tool you're using. So you send the sample information to the instrument control system or the CDS system, and that system then talks to the methods database, and you download the methods information, you send it next door in your company, or you send it outside your organization to a CRO, regardless of the type of instrument you're working with, and you store the methods in a shared corporate database. So we extended the scope last year to also support export of results, so chromatograms and peaks, which is great because that means that we can kind of close the loop for sampling. So in case you have like out of spec analysis, we can use this to look into the reason why the assay is not working. We can use the system now to compare across multiple measurements. So as I said, out of spec or retention time shift, or where you have outliers, or just to see how the system is changing over time. And hopefully next year we are going to do a lot of different use cases to show what we can do with this solution. So what we did in the POC was that we transferred the method parameters between different hardware systems, so different Agilent hardware. We transferred between GSK and Merck, and we also showed that we can accurately and reliably transfer methods between two different CDS systems. And this is the data that the Pankite is now going to show you. I'm going to click here. Pankite, would you like to take control?
Sure. Thanks, Bertie. Can everybody hear me?
Yeah.
Right. Thanks, Bertie, for the introduction to this project. It has been a great effort and significant progress being made through this non-competitive, pre-competitive collaboration of different pharmaceutical companies along with Pistoia Alliance. Azidin unfortunately couldn't make it today, so hopefully I'll do justice to his slides. As shown on this slide, we had three different POCs, concepts, or problem statements. First one was, can we transfer methods across same CDS and same instruments, which is Agilent instruments and OpenLab CDS? Then can we transport those within one pharmaceutical company? If we are able to do that, the next challenge is, can we do the transfer between the same CDS, same instrument vendors across different pharmaceutical companies? And the third challenge was, can we expand the scope further and go beyond a single CDS to different CDSs? So we tackled this at GSK and Merck. So for the first one, what was done was a proof of concept for transferring the methods as well as the data across different Agilent instruments. This work was done by Azidin at GSK, and simultaneously the work was done within Merck, which I'll be talking to later in the slides. Here, what specific objective was, using these ADF concept files, import and export of the instrument method using OpenLab CDS, transfer across 1260 and 1290 instrument, overlaying the data in the CDS cloud system. And these were the successful results obtained. There were five parameters tested, and most of them passed successfully. For some of these issues, some of the aspects, there were some findings which were captured and solved. The second concept, as was said, was transferring the data and the methods from Empower to OpenLab CDS. And I think we can go to the second slide, and Azidin actually recorded the videos. Hopefully they'll play. So here he's trying to show the sequence submission from Zontal to Empower CDS. So all the method information was transferred to Zontal and the sequence was created. You can select the methods which are there in Zontal and as you can see, this was a method that came from partnering organization, which was Merck in this case. And then he's creating a sample ID. Once you submit it, it creates your sequence, which you can select. And then you can import that sequence into Empower. You need to have a template for the instrument that you want to run that sequence on, connected to the Empower CDS, and then it creates that sequence to that template, and it gives message if it is a successful. And then you can see that sequence in the list. You can run that sequence as you can see here, and it's 3 samples, and all the 3 sample information is there, and the method has been selected. You can load that sequence to the instrument and execute it, which is similar of now he's showing the instrument method import, and you're seeing all the parameters were transferred successfully. So this was about sequence submission across different CDSs. The next one is how do we overlay the data in Zontal space? And I'll play the video. So once you have the data collected in OpenLab CDS, you can export the data. The data gets exported to the local folder and then get it swept to the Zontal space automatically. You can program it either to be triggered through user or automatic trigger. And Heiko was of great help while being a part of this POC. The data gets imported, and then you go to Empower. You can export this data from Empower in a similar fashion to Zontal space. This particular integration was worked through Orbis. As you see, it displays messages of what it is doing, and it allows user to an opportunity to change the path if they decide selected the wrong sample set. There is an opportunity to correct here. Once it is exported, it displays that the export is completed. Then you can go to the Zontal space. You'll see that the dataset was imported. So now you have the dataset from Empower and from Agilent, both in Zontal space. And to compare the datasets, you can select the two and go to chromatography viewer. It will show all the injections, and you can select whichever injections you want to overlay. So if I want to add Empower injection later on, there. And as you can see, the results are really good compared to each other, and you can toggle between the different sets selected within the graph. So here we saw how actually the solution works, what all the scenarios were tested from method imports and from data overlays perspective within the same CDSs and across CDSs. Now, in the second part of the presentation, I'd like to talk about specifically two use cases where how the data gets transferred across different companies and briefly talk about the benefits of what we get if we can do this cross CDS comparisons. If I move to the So method portability across different companies, right? What are the benefits? Why are we actually doing this? We just saw that yes, technically it has been made possible using the ADF file formats, but why are companies interested in doing this? The current workflow in the current scenario of method transfers and outsourcing across CROs to different CROs, it has multiple steps. To count, it has 9 to 10 steps in that process where you transfer the method, the receiving lab receives the method, manually creates an instrument method, runs the data, collects the data, create a PDF file, send it to the transferring lab, and then the transferring them lab then does the comparison of the results. It takes a lot of time. There are a lot of manual data transcriptions occurring, and it's costs, and it's a lot of work on the paper and emails. With implementation of digital workflow, we are reducing these number of steps from 10 to 4. And by reducing this, not only by reducing the number of steps, we are improving the workflow efficiency, we are also eliminating a lot of manual data transcription in the paper trail. All of this can be done instantaneously through these electronic systems. Ultimately, what it all translates is to reduction in the analytical method transfer cost. And what are the specific factors or terms that really translates into this reduction of the cost? One is the automation and data integrity. As I said, we are eliminating all the manual steps in the process, and by doing that, we are improving our confidence in the data that it was transcribed correctly. Avoiding downstream out of spec results by reduction of human error. Sometimes, a lot of times, we have a lot of out of specs investigations ongoing just because of some small human transcription error. And most of the people in pharmaceutical industry would be aware of how time intensive and cost intensive that process can be to solve an out of spec investigation in a GMP environment. So by eliminating those manual transcriptions, we are actually downstream avoiding those OS that might have resulted from inhuman error. And last, we are enabling on-demand analytics. Like today, if I transfer this method using this workflow, I'm not waiting for the receiving lab to collect the result and send it to me an email in PDF. And with this globalization, if there is a time difference, there is always this 12-hour or 10-hour time difference. So we don't need to wait for it, as shown by Azadin in the demo, that we could have that overlaid chromatograms instantaneously available for the users to compare. So that's why we are doing this project of automated digital transfers of methods. And talking a little bit about infrastructure, which was briefly explained by Berthe in the introduction slides, is we have a system set up at Merck and a system set up at GSK, which will communicate with the horizontal cloud space. For this particular POC concept, we decided to do it in the public cloud with the generic compounds and methods for the matter of speed to complete this POC. And this can be done even within the private cloud systems with the right amount of controls being installed. So having gone through that, this is a brief summary of what the Merck internal transfer looked like. As you can see, we were able to transfer the methods from Agilent 1290 to 1260 in a fashion similarly shown by Azadin's video, but the results were really comparable. And this transfer was completed within 1 hour of one analyst setting up the instrument and collecting the result on Agilent 1290, uploading the method to cloud system, and then walking up to the another system and downloading it and running it. This is a comparison of the data that was done again within the Zontal space as shown by Azadin's video, where we are able to see that the Merck and the GSK data is comparable. Now you can see that the difference in the data collected in GSK, some of the retention times are eluting later. The benefits of we wanted to go to the identify a factor which is causing this. Now in the conventional workflow, if we do not have those digital tools, I would have written an email to Azadin, hey, this is the difference. We need to start identifying which factor is causing it, and we'll have to go through that stepwise comparison exercises of where the actual differences happen. In this case, what we did was we went into the Zontal cloud space. All the metadata associated with this instrument with this result was there, and we were quickly able to identify which particular instrument parameter or what factor was different. And we identified in this case, it was a dwell volume difference between the two instruments that was causing this difference. So we were able to get to the root cause of this difference, which is still within the acceptable range of system suitability if we would have done a classic method transfer, but still we were able to identify that root cause within 4 hours, whereas a conventional investigation process like this could have taken many more than 2 days easily. Moving forward, method portability across CDSs. We just saw in Azadin's video on how you can do it technically as a solution. Now we'll talk about the benefits. One of the benefits that we foresee from this kind of automation procedure is productivity increase and improved instrument usage. Now, we are not having a certain instrument restricted to a certain CDS. We can use any instrument connected to any CDS for this. So and we all know that in pharmaceutical companies, a lot of instruments are connected to different varieties of CDSs. So by allowing this automated transfers, we are not restricting the use. Moreover, if we are decreasing the number of out of specs results downstream, whenever there is an out of specs result in a GMP environment, you need to keep that instrument not being used for any other projects till that out of specs result gets solved. Now, by eliminating the number of out of specs results or by speeding the time about how you can get to the root cause identification, we are actually reducing the amount of time that instrument that cannot be used. So that's also translates into an improved instrument usage. And we are not sometimes as a scientist working in the lab, I have done it in my life that I will enter the method information incorrectly because of human error, and then I'll have to rerun that sample set completely with the right method. Now, if we are eliminating that human error and every time the method being run is correct, we do not need to rerun the sample sets, so that also translates into improved instrument usage. The second benefit is data harmonization and central storage. Since all this information is in one place, we can have method lifecycle simplification process easily mapped out. We can do control charting of the data, how our method is performing over a period of its time, how a specific instrument is performing over a period of time. Second is data access for regulatory and QA/QC. In some of these videos, you had seen that the software prompts messages that the sample set is complete. Do you want to proceed further? There were steps where we saw error messages being displayed. If two instruments, for example, 1260 and 1290, have different pressure limits, then it will prompt you a message that this pressure limit, that method that you're trying to run on this instrument doesn't have the required pressure limit. Do you still want to run it? That will help you capture that command. That gives the flexibility to the user to accept or reject it. But what it does is, whatever the user does, it captures it in the audit trail. So we are trying to it's automatically creating a QA/QC trail list. And another thing is enabling on-demand analytics. As I said, we can do control charting, method lifecycle management, and other aspects of analytics that we want to do because all the data is stored in one place. So for this particular exercise, what we did was within Merck, we transferred the method from an Agilent 1290 binary controlled by Open Lab CDS. And you'll see there was this, we were very selective in kind of instruments that we were selecting for the reason that we wanted to capture the variability. We wanted to see if this solution doesn't get bottled down because of the instrument variability. We took this method from a 1290 binary to a 1290 quaternary controlled by Empower CDS. So there are two differences here. The CDS is different and the instrument configuration is different. So first thing is we were able to successfully transfer the method. Transferring the method from binary to quaternary, it says, oh, you are trying to run this method on a quaternary system. And I, as a user, I accepted that, yes, I want to run it because technically it should be able to run it. And I got the following results. Now, as you can see in the results, the retention times are shifted for a quaternary system. So we started thinking about what could be the cause of this differences in the retention time. And ultimately, it was again transferred into the dual volume. That the dual volume of a quaternary instrument is higher than that of the binary instrument. And when we did the calculations, the differences in the dual volume translated to the differences in the retention time. Again, talking to the point that it allows you to do on-demand analytics and identify the root cause of the problem. So to conclude, through this method portability, we have shown that we can transfer the methods across different instrument vendors, across different CDSS systems, and across different pharmaceutical companies. And all this was enabled because we had that standardization of the data formats in the form of allotrope data format. To talk about where the project started and to where it is today, it started back in 2018 and 19, where Pistoia Alliance developed a method model governed by allotrope. The efforts then was focused on building architectural solutions. Thereafter, in 2020 and 2021, some of the testings were being implemented. Initially, Merck and AbbVie partnered to implement, to test the first POC. Then the building of method ADF for Agilent hardwares and Empower was focused in the second part of all of 2021. In 2022, Q1, Q3, Merck and GSK collaborated to test this new POC with this new ADF builds available. And in the remaining part of this Q4 of 2022, we are extending the instrument coverage to Waters hardware with help from Orbis. That is helping us build those Empower adapters. So we have successfully shown that it works within Agilent systems, Agilent instruments connected to Agilent or Empower CDSs. Now the team is focusing on extending the coverage of the hardwares to Waters. Call to action. These are the benefits. We have shown through these two POCs that this is a technical solution that can be actually implemented in the real world. So we are asking other pharmaceutical companies to join the implementation plans, join these efforts, and this is your chance to influence and the direction that this team takes in moving forward. If there are any specific needs that are required for your organization, you can join the Pistoia Alliance group. Waters hardware extension has started, and we will be completing it by end of this year. We are extending for the next year. The projects could be extending the solutions to cover CDSs from different vendors. We will have Empower and Agilent now, but there are other CDSs. And next year, more efforts will be directed towards simplified automated data analytics and visualizations. The visualizations that you saw within the Zontal space, we'll be working with them more to build more advanced data visualizations within their platform. And again, thanks to Pistoia Alliance for managing this project, and Azadin from GSK to contribute and work on this project. I'm sure we'll be happy to take any questions.
Great, thank you both.